Place your hand on any part of your body, your arm, your chest, your face. What you're touching isn't just tissue. It's a vast network of protein machines in constant motion. Every cell in your body is busy breaking down and rebuilding proteins right now, dismantling and reconstructing itself piece by piece. That chicken breast on your plate isn't going to become muscle directly. First, it must be completely disassembled, broken down into individual amino acids, its very structure dissolved. Then your body performs a molecular miracle. It takes these basic building blocks and reconstructs them into uniquely human proteins, following genetic instructions written in your DNA. You are, quite literally, what you eat, but not in the way you might think. Your body is constantly harvesting amino acids from your diet, mixing them with recycled proteins from your own tissues, and rebuilding itself according to its precise blueprints. Every heartbeat, every breath, every thought depends on proteins working in perfect coordination. This isn't just nutrition, it's the chemistry of life itself. Join me as I reveal how your body performs this remarkable feat of molecular engineering, transforming the proteins you eat into the living machinery that makes you who you are. This is Food Truth, the Protein Show. Take a piece of silk fabric between your fingers. Feel its complex structure, threads woven into an intricate pattern. Now watch as we begin to unravel it, strand by strand. This is how your digestive system approaches protein, a methodical deconstruction of something complex into its basic elements. Place a piece of chicken in your mouth. As you chew, your teeth are starting what pepsin in your stomach will finish, breaking bonds that hold protein chains together. Feel your tongue move against your palate. Those mechanical forces are the first step in dismantling protein's architecture. Touch your stomach. Inside, hydrochloric acid is creating the perfect environment for protein-cutting enzymes. Like molecular scissors, they snip long protein chains into smaller and smaller pieces. What was once muscle fiber becomes peptides, then individual amino acids, the letters of the protein alphabet. These amino acids flow into your small intestine, where specialized transporters wait. Each one passes through your intestinal wall like a key finding its lock. They enter your bloodstream, but they're not staying there long. Your liver has plans for these building blocks. Place your hand on your right side, just under your ribcage. Your liver is performing a molecular miracle, reassembling these amino acids into new proteins. Some will become enzymes to drive chemical reactions, while others will form antibodies to fight infection. Still, others will build structural proteins for your muscles, skin, and bones. This isn't just one assembly line. It's thousands working in parallel. Your liver is reading genetic instructions, linking amino acids in precise sequences. Like a biological 3D printer, it's crafting proteins unique to your needs at this moment. That chicken protein isn't becoming human muscle directly. It's being completely rebuilt, letter by letter, into human proteins. Watch these new proteins leave your liver, each headed to fulfill its destiny. Some race to repair microdamage in your muscles, others join the endless patrol of immune proteins in your blood. Still, others become hormones, carrying messages to distant cells. This constant deconstruction and reconstruction never stops. Every protein in your body will eventually be broken down and rebuilt. You are, in a very real sense, constantly reassembling yourself from the proteins you eat, taking apart foreign proteins and rebuilding them into you. The story of protein digestion is a masterpiece of chemical precision. Every step is carefully choreographed, from the moment pepsinogen transforms into active pepsin until the final amino acids are absorbed. This isn't just digestion, it's a biochemical cascade that evolved over millions of years. Your stomach's parietal cells maintain a pH of around 1.5 to 3.5, acidic enough to denature proteins, but not so acidic that it destroys the enzymes needed to digest them. This acid bath serves multiple purposes. It unfolds proteins from their complex three-dimensional structures, activates digestive enzymes, and creates the perfect environment for protein breakdown. The star of this acid environment is pepsin, but it starts as an inactive form called pepsinogen. This is crucial. You don't want protein digesting enzymes active until they reach the stomach. The stomach's acid triggers pepsinogen to clip off a piece of itself, transforming into active pepsin. It's like a molecular switch being flipped, turning on the protein cutting machinery. Pepsin has a specific job. It cuts proteins at particular amino acid bonds, preferring to slice between hydrophobic amino acids. But it's not working alone. As proteins unfold in the acid environment, they expose more sites for pepsin to access. This creates a feedback loop. The more acid denatures the protein, the more efficiently pepsin can cut it. The result isn't individual amino acids yet. Instead, pepsin produces peptides of varying lengths. These partially digested protein fragments then move into the small intestine, where they meet a new cast of digestive characters. The pancreas contributes trypsin, chymotrypsin, and carboxypeptidases, each with its own protein-cutting specialty. This coordinated enzyme attack is remarkably efficient. Each enzyme has evolved to recognize specific patterns in protein chains. Trypsin cuts after positively charged amino acids. Chymotrypsin prefers bonds after large hydrophobic amino acids, while carboxypeptidases work from the ends inward. Together, they reduce proteins to smaller and smaller pieces. But even this isn't the end. The small intestine's brush border contains aminopeptidases that complete the breakdown into individual amino acids. These enzymes are embedded in the intestinal wall itself, ensuring that the final steps of protein digestion happen right where absorption will occur. The timing of this process is crucial. If proteins were broken down too quickly, the flood of amino acids would overwhelm transport systems too slowly, and nutrients would be lost. The sequential nature of digestion, from stomach to small intestine to brush border, creates a perfect gradient of breakdown. What makes this process remarkable is its adaptability. The system can handle different types of proteins, from the complex structures in meat to the simpler ones in legumes. The acid-enzyme partnership can denature and digest virtually any protein you consume, reducing it to components your body can use. Understanding this chemical ballet helps explain why protein digestion can be disrupted by various factors. Acid-reducing medications can impair the initial protein denaturization. Pancreatic problems can reduce enzyme availability. Celiac disease can damage the brush border where final digestion occurs. Each step in this precise chemical dance matters. Every protein in your body is a molecular masterpiece, built from a set of just 22 amino acids. But comparing this to having only 20 letters in an alphabet misses the true complexity. It's not just the sequence that matters, it's how these chains fold and interact in three-dimensional space that brings proteins to life. At the most basic level, each amino acid connects to its neighbors through peptide bonds, specific chemical links between the carboxyl group of one amino acid and the amino group of the next. These bonds aren't random. They form at precise angles, creating what we call the primary structure. This backbone provides the foundation for everything that follows. The chain begins to fold almost immediately, driven by the chemical properties of the amino acids themselves. Some are attracted to water, others repel it. Some carry positive charges, others negative. These interactions drive the formation of local structures, alpha helices spiraling like molecular springs, and beta sheets folding like molecular pleats. This is secondary structure, and it's essential for protein function. At the local structure's form, they interact with each other. Helices and sheets arrange themselves into specific three-dimensional shapes, driven by chemical forces between distant parts of the protein chain. This tertiary structure is what gives each protein its unique shape and function. It's why hemoglobin can carry oxygen, why antibodies can recognize invaders, why enzymes can catalyze specific reactions. But many proteins don't stop there. Individual protein molecules can come together to form larger complexes, quaternary structure. Hemoglobin, for instance, consists of 4 protein chains all working together. These assemblies aren't random. They're precisely controlled by the same chemical forces that drive the folding of individual chains. The process of protein folding is remarkably precise. Chaperone proteins help ensure that newly synthesized proteins fold correctly. Quality control systems identify and destroy misfolded proteins. This precision is crucial because misfolded proteins can lead to diseases like Alzheimer's or Parkinson's. Water plays a crucial role in this architectural marvel. Hydrophobic amino acid clusters together in the protein's core, avoiding water contact. Hydrophilic amino acids on the surface interact with water, helping maintain the protein's shape. This arrangement is so precise that changing even a single amino acid can sometimes disrupt the entire structure. Disulfide bonds, which are strong chemical links between specific amino acids, can lock parts of the protein in place. These covalent bonds act like molecular staples, maintaining crucial aspects of protein shape. They're particularly important in proteins that must maintain their structure in harsh environments, like digestive systems. Post-translational modifications add another layer of complexity. Proteins can be modified after synthesis by adding chemical groups, phosphates, sugars, lipids, etc. These modifications can change how the protein folds, how it interacts with other molecules, or where it goes in the cell. Understanding protein structure is crucial because it determines function. From the enzymes that digest your food to the antibodies that fight infection, from the collagen that gives structure to your skin to the ion channels that allow your nerves to fire, every protein's function depends on its precise three-dimensional architecture. The journey from your intestinal lumen to your bloodstream isn't a simple passage. It's a sophisticated transport operation involving multiple carrier systems, energy gradients, and cellular checkpoints. Each amino acid must pass through a gauntlet of precisely controlled barriers before it can be used by your body. At the intestinal brush border, specific transport proteins await their cargo. These aren't passive channels. They're highly selective carriers that recognize specific amino acid structures. The BO system handles neutral amino acids, while the BO plus system manages basic amino acids plus some neutral ones. Each system has its own specificity and capacity. Many of these transporters work through sodium cotransport, riding the electrochemical gradient that your cells maintain. As sodium moves into the cell down its concentration gradient, it pulls amino acids along with it. This process, called secondary active transport, allows cells to absorb amino acids even against their concentration gradients. Small peptides follow a different path. The PEPT1 transporter can move and die and tripeptides intact across the intestinal membrane. This system is particularly efficient, allowing for rapid absorption of small protein fragments before they're broken down into individual amino acids inside the cell. Competition between amino acids for transport creates a complex dance at the absorption surface. Large neutral amino acids like leucine, isoleucine, and valine share transporters and compete for absorption. This is why massive doses of single amino acids can actually impair overall protein absorption. They monopolize the transport systems. Once inside the intestinal cells, amino acids face another challenge: getting out. The basolateral membrane, facing the bloodstream, contains different transporters from the brush border membrane. Systems L, Y+, and A manage this exit process, carefully controlling which amino acids enter circulation. The liver acts as a primary checkpoint for newly absorbed amino acids. Through first-pass metabolism, it can remove up to 60% of certain amino acids from portal blood, regulating which ones reach the general circulation. This processing helps maintain amino acid homeostasis throughout the body. Absorption isn't uniform along the intestinal tract. Different regions showing varying concentrations of specific transporters. The jejunum, with its extensive brush border, handles the bulk of amino acid absorption, while the ileum serves as a backup system to catch any remaining nutrients. Hormones and metabolic signals constantly modulate this absorption process. Insulin increases amino acid uptake in many tissues, while glucagon has opposite effects. Even the transporters themselves can be regulated, with their numbers increasing or decreasing based upon dietary protein intake. The efficiency of this system is remarkable. Under normal conditions, less than 2% of dietary protein escapes absorption. This high efficiency comes from the coordinated action of multiple backup systems and the ability to absorb both amino acids and small peptides through different pathways. Before any enzyme can begin its work, proteins must be made accessible. Mechanical digestion isn't just preliminary. It's a sophisticated process that transforms complex protein structures into particles small enough for chemical digestion to be effective. This physical breakdown begins the moment food enters your mouth. Your teeth are specialized tools. Each type evolved for a specific aspect of mechanical breakdown. Incisors slice through protein fibers, while canines tear and shred complex structures, while molars grind and crush. This mechanical action doesn't just reduce food size, it disrupts the quaternary and tertiary structures of proteins, beginning the process of denaturization. The tongue performs a crucial choreography during this process. It doesn't just move food around randomly, it systematically positions food between the teeth for optimal breakdown. This movement, combined with the mechanical forces of chewing, creates a precise pattern of physical disruption that maximizes surface area for later chemical digestion. Saliva, while not directly breaking down proteins, plays a vital role in mechanical digestion. It lubricates food particles, allowing them to be formed into a bolus, which is a coherent mass that can be safely swallowed. The moisture also begins to separate protein fibers, making them more accessible to upcoming digestive processes. As the bolus moves through the esophagus, peristalsis, which are coordinated muscle contractions, provide additional mechanical force. These waves of contraction don't just transport food, they continue the physical breakdown process, applying pressure that further disrupts protein structures. The stomach's mechanical action is particularly sophisticated. Its muscle walls create three distinct types of movements: mixing waves that churn the contents, propulsive waves that move food towards the pylorus, and grinding actions that physically break down protein structures. This rhythmic churning can generate forces up to about 0.2 of a newton, strong enough to disrupt most food matrices. The rate and force of these contractions are precisely controlled by mechanical receptors in the stomach wall. These sensors detect both the volume of food present and its physical resistance to breakdown, adjusting the stomach's mechanical activity accordingly. This ensures optimal physical processing before chemical digestion begins in earnest. The physical disruption of protein structures serves another crucial purpose. It exposes hidden antigenic sites that the immune system needs to evaluate. This is part of your body's sophisticated system for identifying potentially harmful proteins, a process that requires mechanical breakdown to be effective. The efficiency of mechanical digestion directly impacts the effectiveness of chemical digestion. Inadequate chewing or reduced stomach motility can significantly impair protein breakdown, even if chemical digestion is functioning normally. This is why proper chewing is crucial for optimal protein absorption. Temperature also plays a role in mechanical breakdown. The heat in your mouth and stomach often helps soften protein structures, making them more susceptible to physical forces. This thermal effect complements the mechanical forces, preparing proteins for the chemical phase of digestion that follows. Your liver isn't just another stop on the protein processing journey. It's the command center for protein metabolism. This remarkable organ receives about 80% of its blood supply from the portal vein, giving it first access to nearly everything absorbed from your intestines, including amino acids from protein digestion. When amino acids enter the liver, they face their first major sorting decision. Some will be used immediately for protein synthesis, while others will be modified or broken down, and some will be released back into circulation for use by other tissues. This triage process involves sophisticated chemical sensing mechanisms that monitor both incoming amino acids and the body's current needs. Diminution is one of the liver's crucial protein processing functions. When amino acids are broken down, their amino groups must be removed. The liver contains specialized enzymes that perform this task, converting the amino groups into urea through the urea cycle. This process prevents toxic ammonia buildup while allowing the carbon skeletons to be used for energy or converted to glucose or fat. But the liver doesn't just break proteins down, it's also the body's primary protein synthesis factory. It produces most of your plasma proteins, including albumin, which maintains proper fluid balance in your blood, and clotting factors essential for wound healing. These proteins are assembled according to precise genetic instructions, then packaged and released into the bloodstream. Transamination represents another sophisticated aspect of the liver's protein processing. Through this process, the liver can remove amino groups from some amino acids and attach them to others, effectively converting between different amino acids as needed. This ability to remix amino acids helps maintain optimal amino acid balance throughout the body. The liver also serves as a short-term amino acid storage system. While proteins can't be stored in the way that fats and carbohydrates can, the liver maintains a crucial pool of amino acids that can be quickly accessed to meet sudden demands. This pool is constantly being depleted and replenished as the body's needs change. Gluconeogenesis, the production of glucose from non-carbohydrate sources, relies heavily on the liver's proteins processing capabilities. When carbohydrate intake is low, the liver can convert certain amino acids into glucose, helping maintain blood sugar levels. This process is carefully regulated to preserve essential amino acids while still meeting energy needs. The acute phase response, a critical part of your immune system, depends on the liver's protein synthesis capabilities. During infection or injury, the liver rapidly shifts its production priorities, increasing output of defensive proteins while decreasing production of less immediately essential proteins. This metabolic flexibility is key to survival during stress or illness. Quality control is another crucial aspect of the liver's protein processing role. Misfolded or damaged proteins are identified and either reprocessed or marked for destruction. This system helps prevent the accumulation of dysfunctional proteins that could harm cellular processes. The liver's protein processing capabilities also include detoxification of protein-related metabolites. Excess amino acids, which can be toxic if allowed to accumulate, are processed through specific pathways that either incorporate them into useful molecules or prepare them for safe excretion. This cleanup role is essential for maintaining metabolic health. Inside every cell, protein synthesis represents one of the most complex manufacturing operations in nature. Ribosomes, molecular machines composed of both protein and RNA, serve the assembly platforms where individual amino acids are linked together in precise sequences. Each ribosome can assemble proteins at a rate of 6 to 9 amino acids per second. The process begins in the nucleus, where specialized enzymes unwind specific sections of DNA. RNA polymerase reads these genetic instructions, creating messenger RNA, or mRNA, through a process called transcription. This mRNA serves as a mobile blueprint, carrying assembly instructions from the nucleus to the ribosomes in the cytoplasm. Before protein assembly can begin, each amino acid must be activated and prepared for incorporation. Transfer RNA, or tRNA molecules, shaped like molecular clover leaves, are charged with specific amino acids by aminoacyl tRNA synthetases. These enzymes are remarkably precise. They almost never attach the wrong amino acid to a tRNA. Translation begins as ribosomes bind to mRNA molecules. The ribosome reads the genetic code 3 letters at a time, with each triplet of nucleotides, the codon, specifying a particular amino acid. This reading frame must be maintained perfectly. A shift of even one nucleotide would result in a completely different protein sequence. As the ribosome moves along the mRNA, it facilitates the formation of peptide bonds between amino acids. Each amino acid is positioned precisely, then chemically linked to the growing protein chain. This process requires energy in the form of GTP, ensuring that only correct amino acid additions can occur. Quality control systems operate at multiple levels during synthesis. If the wrong amino acid is accidentally incorporated, or if the protein begins to fold incorrectly, specialized proteins recognize these errors. Some proteins can even be removed from ribosomes mid-synthesis if serious errors are detected. Post-translational modifications add another layer of complexity to protein synthesis. Many proteins undergo chemical modifications after their initial assembly. Phosphate groups are added or removed, sugar molecules are attached, or specific amino acids are modified. These changes can dramatically affect a protein's function and location within a cell. Protein folding begins even as the chain is being synthesized. Chaperone proteins assist in this process, preventing inappropriate interactions and ensuring proper folding. Some proteins must fold into precise shapes immediately, while others require additional processing in the endoplasmic reticulum or Golgi apparatus. The energy cost of protein synthesis is substantial. Cells spend approximately 75% of their ATP on protein synthesis and related processes. This high-energy investment reflects both the importance of proteins and the complexity of their assembly. Each peptide bond formation requires at least 4 ATP molecules. The entire process is tightly regulated in response to cellular needs. When amino acids are scarce or when cells need to conserve energy, protein synthesis can be rapidly downregulated. Conversely, certain signals like hormones or growth factors can dramatically increase protein synthesis rates in specific cells. Your skeletal muscle is in a constant state of flux, simultaneously breaking down and rebuilding itself. This isn't inefficiency. It's a precisely controlled process that allows muscles to adapt to changing demands. Every day, your body breaks down about 300 to 400 grams of protein, much of it from muscle, while simultaneously rebuilding these tissues. The breakdown process, called catabolism, involves several sophisticated protein degradation systems. The ubiquitin proteasome pathway tags specific proteins for destruction, while autophagy engulfs and digests larger chunks of cellular material. These systems increase their activity during fasting, illness, and after periods of disuse, leading to muscle loss. Mechanical tension during exercise triggers anabolic signaling through mechanicotransduction. Physical stress is converted into chemical signals. These signals activate mTOR, or mammalian target rapamycin, a master regulator of protein synthesis. When mTOR is activated, it increases protein synthesis in muscle cells, particularly of contractile proteins like actin and myosin. Amino acids, especially leucine, play a crucial role in this process. Leucine not only serves as a building block for new proteins, but also acts as a signaling molecule, directly activating mTOR. This dual role explains why leucine-rich proteins are particularly effective at stimulating muscle growth. Hormones orchestrate this balance between breakdown and growth. Insulin suppresses protein breakdown and promotes synthesis, while glucagon and cortisol increase breakdown. Growth hormone and testosterone promote protein synthesis and muscle growth, working through multiple signaling pathways. Satellite cells, muscle stem cells lying dormant between muscle fibers, activate in response to damage or intense exercise. These cells divide and fuse with existing muscle fibers or form new ones, contributing to muscle repair and growth. This process requires precise timing of protein synthesis and cellular fusion events. The energy cost of maintaining muscle mass is substantial. Each kilogram of muscle tissue requires about 13 to 15 kilocalories per day just for basic protein turnover. This high energy cost explains why the body readily breaks down muscle during periods of energy restriction. It's an expensive tissue to maintain. Resistance exercise creates microscopic damage to muscle fibers, triggering a repair response. This isn't just damage control, it's an adaptive response that results in stronger muscle fibers through increased protein synthesis. The repair process typically peaks 24 to 48 hours after exercise, explaining delayed muscle soreness. Age affects these processes significantly. The anabolic response to protein intake and exercise becomes blunted with age, a condition called anabolic resistance. This requires older individuals to consume more protein and exercise more deliberately to maintain muscle mass. The integration of these breakdown and growth processes allows muscles to remodel themselves based on demand. Regular exercise, adequate protein intake, and proper rest create an environment that favors muscle protein synthesis over breakdown, leading to maintenance or growth of muscle mass. Well, that does it for this episode of Food Truth. Thanks for joining me as we took our deep dive into the world of protein. I hope you join us next time as we take on the somewhat cryptic and mystic subject of processed foods. We're actually going to be talking about how processed foods really affects your body and what it's doing inside you after that first bite. So I hope you join us next time. If you ever want more Food Truth, go to our website,